and we have learned new ways of doing research and seeing how technology has advanced over the years. This session is a final session, and it will be in two parts. The first part will be strictly practical, which is intro to relevant databases for virtual screening. And the second part will be both a lecture and a practical, which is a structure-based virtual screening, also known as molecular docking. So I'll be the anchor for the first part, and I will introduce our speaker, facilitator for this session, which is Mr. Opeyemi Iwaloye, and he's going to be talking about intro to relevant databases for virtual screening. So Mr. Opeyemi Iwaloye is a graduate from the Department of Biochemistry at Adekunle Ajasin University in Nigeria, where he had his BSc, and he got his MSc from Federal University of Technology, Akure, Nigeria. He currently works in Bioinformatics and Molecular Biology Laboratory, Federal University of Technology, Akure, and Federal Polytechnic Ado-Ekiti, respectively. He has not less than 7 years of cogent experience in structural bioinformatics and molecular biology, and has participated in numerous research projects, which have yielded 34 scholarly articles. He recently won Blazer Fellowship with the University of Alabama at Birmingham University, and will start his PhD in biochemistry and structural biology by fall 2024. His current area of study is identification of drug-like molecules from natural origin, which can modulate signaling pathways associated with the onset of diseases, including cancer, diabetes, and SARS coronavirus 2. Join me as I welcome Mr. Opeyemi as he takes the stage and gives us his talk. You're welcome.
Thank you for the introduction, and yeah, so good afternoon, everyone. My name is Opeyemi, and today I'll be taking you through the relevant database that are associated with molecular docking. So yeah, today I will just be sharing my screen with you before we start. So okay, this is my updated profile. Like, I'm currently a PhD student at School of Medicine, University of Alabama at Birmingham, and of course I have 7 years, more than 7 years experience, and my research interest is focus on currently is focused on brain tumor therapeutics and computer-aided drug design, and I have been able to publish a quarter series of research over the years, and so far those research have accumulated up to 640 citations based on Google Scholar metrics. So it's very nice to speak with you today. So I'll be taking you through the database relevance to molecular docking study. So the first question here is there a need for biological database? So absolutely, everyone will say yes, there is. So then why is database so important for molecular study? So we all know that the major objective of those databases is just to organize data that are collected by different scientists from different fields, so that its content can be easily be accessible, managed, and updated. So also we also know that the reason why there is need for these databases, over the years there has been an increase in the retrieval of data-rich sciences, and those data can come from from the biologicals or biomolecules, which may include the protein sequences, the nucleotide sequences, of course, the 3D data structure, and the 2D structure of of biomolecules, such as compounds, which includes can be synthetic or natural compounds. So storing this data enable us to understand the structure and function of the biomolecules. And you can understand the structure and function of biomolecule using the QSAR model, and this enable us to also like index our data and remove redundancy of data as well. So so why are these what are the database relevance to molecular docking study? So there are a couple of database that are relevant to the molecular docking, and this includes the protein database, and this database contains the 3D structure of a proteins that has been crystallized using X-ray crystallography or other structural biology techniques. Also, this you know we also have the protein sequence database. So this database includes uh, repository where you can retrieve the protein of sequence of proteins from diverse microorganisms, including human as well. So also we have a database for modeling, for protein modeling. So in this case, there are some, for example, there are some proteins that the 3D structures have not been co-crystallized, and in this case, we have to model the protein by by trying to find similar templates of those proteins based on the known sequence of the protein. Also, we also have the protein that compound database. So this database is where we can retrieve compounds like the synthetic compounds, the natural compounds. So we have diverse compound database, and also we have a database for evaluating the toxicity of compounds. So for example, we have compounds, we've identified elite compounds using molecular docking studies, and we want to like evaluate uh, the drug-like properties and the toxicity. So we can use this database. So this database use they actually use like it's like a machine learning based method where they train some data sets to predict the bioactivity or the pharmacological activity of a compound. So we also have a lot of other databases, but these databases are major database that are relevant to molecular docking study. So now we now dive into the protein database. So the protein database that contain one or more data set about proteins, which may include the protein amino acid sequence, the conformation of the protein, the structure, the certain features like the catalytic site, the active sites of the protein. Also, this database are compiled by translation of DNA sequence from different gene databases and structure information. And they're important resource because we all know that proteins mediate most biological functions. So one of the most famous protein database is PDP, which is the protein data bank. So this protein data bank is like a reservoir of larger compound, like larger proteins, like larger micro biomolecules, and this PDP contains three-dimensional structure of molecules, and this entails the proteins, the nucleic acid, and in this database we can find a lot of proteins which contain up to like 150,000 proteins, and also nucleic acid and protein nucleic acid complex. So how are these proteins how are these proteins deposited in the PDB, the protein data bank? So these proteins are derived, like the information from this protein are derived from three different sources. And this includes the X-ray, crystallography, the LMR, and the electron microscopy. So this shows the numbers of the proteins that are deposited in the protein database or data bank over the years. You can see that there's an increase in there's increase in the awareness of structural biology. There's increase in the deposition of these protein structures in the in protein data bank over the years. So we also have other relevant protein database, and this includes the PubMed. I'm sure you have explored, most of us have explored PubMed. So, and PubMed contains over 5 million records of, scientific informations like the compound's name, the compound, the bioactivity of the compounds or compounds against a specific cell line. Also, we have the UniProt. So the UniProt is also a very nice database where you can retrieve high quality and free accessible resource of a protein sequence and functional information. So in this database, you can get the faster sequence of the protein. You can get information about a protein, which includes the amino acid residues, like the sequence. You can get the active sites of the protein, the catalytic sites. You can get a lot of information about. Then you can also get the mutated form of the protein as well, like the sequence of the mutated form of the protein. So also we also have the ChemView database. I often use this ChemView database to retrieve information of compounds that have been with known biological activities. For example, scientists use compounds, either synthetic or natural compounds. They test the pharmacological activities of the compounds against a specific cell line in vitro, and they measure the bioactivity based on IC50, uh, uh, IC50 or EC50. So this database could be used to get information about compounds with activities that has been used against a particular proteins and the likes. So also I made mention of the protein, the modeling database. So this includes these are database where you can model your proteins. For example, if the structure of the protein has not been deposited on protein data bank, so you can use protein modeling to model your protein. So and it has become necessary to construct a model of proteins when the 3D structure is unknown. So example of database that can be used to um, predict the 3D structure of a protein is Grow Here. So Grow Here has been over the years. Grow Here has been shown to is a database has been shown uh, to accurately predict accurately predict the 3D structure of a protein of interest based on sequence of the protein. So we also have a lot of database for modeling of protein. We have the uh, we have the Pyrex, we have the Sage models, we have the Swiss model, we have the Orion. So this database has different large machine learning based method that were used to train, the uh, the templates so that they will be able to be able to uh, accurately predict the 3D structure of protein. So among this database, there's no study that have shown that one of them is the best. So you might use two different database. For example, if you want to model your protein, you can use two different database and uh, confirm which one has the best has model the best structure based on the it could be based on the missing side chain among others. So we also have the compound database. So this compound database is used to predict to retrieve the 2D structure of proteins. And if you want to conduct molecular docking uh, after you've identified your target, then you need a molecular in form of ligand to target your proteins, proteins. And in this case, you need like a compound, like a peptide. Uh, so definitely you are going to need a compound to dock your protein. So we have a lot of compounds database. We have the PubChem, which is the world's largest collection of free accessible chemical information and contains pure characterized chemical compounds, and the database is maintained by National Center for Biotechnology Information. So you can retrieve a lot of compounds from here. And also, we also have the Ken Spider, which is another fantastic compound database where you can get a lot of information about a compound of interest. For example, let's say you've docked a compound and you found out that it has high docking score against a protein, and you want to uh, conduct further analysis. So you can come to the Chem Spider to check about the chemical supplier of that compound. So and this will enable you to like request and get the compound. So you can find a lot of information about the compounds on the Chem Spider database as well. So then we also have other compounds database. So those the Chem Spider and the PubChem contains both natural and synthetic compounds. But there are some database that are specifically for a unique set of compounds, like the natural compounds. So, for example, we have the NPAS database. I think I use this database couple of times for virtual screening. So here you can retrieve as much as 500,000 compounds from natural sources. So here you can just, for example, you are trying to identify the lead compounds against a target. Let's, for example, insulin receptor. So you can just dock all these 470,000 compounds against these targets and identify the lead compound. So from there, you can subsequently streamline the lead compounds with using the admin properties among others. So we also have a database for we have the TCM database, which is a Taiwan database. So this database also contains more than 20,000 compounds derived from Chinese traditional medicine. So also we have database to predict drug likeness and toxicity. We have the ADMESA and also the Swiss ADME. So we have a lot of database that you can use to predict your compound drug likeness and toxicity. And these two are just uh, two of so many database out there. So so then so the next section is I'm going to just introduce you or take you through some of those database that are relevant to molecular docking study. So the first most important database for the protein database is the PDP. So PDP, after you've had like experimental design, before you like do molecular docking and the likes, you would have had a plan, like you have hypotheses and you want to test the hypothesis. So it's just like the wet experiment. We have to get reagents. You have to so you have to get like compounds, get things you want to use. So for in silico study, the first you have to retrieve is the compound, the protein. So you have to be sure that the protein is on has been the 3D structure of the protein has been characterized using uh, either of the structural biology methods. So then after that, you go to PDP, which is data protein data bank, to retrieve the protein. So I'll be taking you through the protein data bank. So so before I proceed, sorry, I think I'm not so organized with this presentation. I mistakenly thought my session is on Monday, so I just got to know about 15 minutes ago. So, but that's fine. I've done this couple of times. So, protein. So we have to go to protein data bank. Data bank. So, uh, so protein data bank. So we just have to type protein data bank on Google. Then the first entry is the home page to protein data bank. So we have to click on this first link. And okay, so let me just assume that hypothetically I want to my research is focused on diabetes. Yeah, diabetes. So I want to work on diabetes, and I've actually identified that I've actually I have some plants like based on literature search, I I found out that the whole extract of Chromonella odorata, which is a plant, have a biological activities is against the is able to reduce blood glucose level. So, but you understand that this plants extract contains thousands of phytochemicals, right? Yeah. So what I want to my aim is just to identify which of those compounds is able to effectively bind to insulin receptor. So and based on research literature search, I've been able to identify a lot of compounds that have been isolated from this Chromonella odorata. And so right now I have the compound, and so I want to use the compounds to target the protein, right? And the protein is insulin receptor. So what I'm going to do is so to search the insulin receptor, we can either go to uh, literatures, like we can either go to literature like previously published data on insulin receptor and look out for proteins that has been used for molecular, like proteins peculiar to insulin receptor that has been used for molecular docking study. Or we can just come here and just type insulin receptor. So this is the search box, insulin. So we can type insulin receptor, right? Receptor, and Homo sapiens. So uh, so not much. So insulin. So let's type insulin receptor. Okay. So it has given us list of insulin receptor based on different organism. You can see we have Drosophila. We are interested in the insulin receptor of Homo sapiens. So we can streamline the search by just scrolling down and so we can streamline the search by just clicking Homo sapiens and click on this. So we click on Homo sapiens and this. So based on the what I click, we can see the structural basis for the insulin inhibition of inhibition of insulin-like growth factor by IGF binding protein. So depending on what we want, but what I want you to take a look at, like what is more important is what you have to understand, that you have to have it in the back of the mind, the name of the protein you are looking for. That is the first thing. Then second thing, you will look for uh, the organism. So which organism are you using? Is it Homo sapiens? So for example, if you are like working on in vivo experiment, like you've treated your, let's just say this saponin fraction of this Comonella, you've actually administered it to the mice, and you found out that it has decreased insulin glucose level, right? So you can just in this case you can use, because your model is rats, so you can try to use rats. So the species might be rats. So if it is rat, then that means it is Ratus novegicus or Ratus. So it depends. So but in this case, we're interested in interested in Homo sapiens. So we can uh try to look for the insulin receptor using like insulin receptor. So another thing I want you to take note is so once you find the structure, you also have to check if this insulin receptor is in complex with something. So it has to be in complex with maybe a molecule or a protein, like in complex with a protein. So in this case, if it is not in complex with a protein, that means the active site of the protein is not identified, it has not been defined, because it's kind of very, very important for you to pick a protein that is in complex with a molecule, because the molecule is where the is where the active site of the protein is. So it's very, very important. So let me just, because of time, let's just assume this is the insulin receptor, what we are looking for. So then we also have to check what is the method that is being used. So it is X-ray diffraction. What is the resolution? The 2.5 Armstrong. So the lower the Armstrong, the better the quality of the protein. So this Armstrong is used to define the resolution of the protein following, following the crystallization, like the characterization. So the lower the Armstrong, then the better the quality of protein. So it's very important to also take this into consideration. I think anything below 3.0 is fine. But if you didn't see the protein with 3.0, less than 3.0 Armstrong, then you can just pick that as well. But make sure you prepare the protein uh, because that's very, very important. So the higher the value of the resolution, then the lower the quality of the protein. So then after this, we just click on the structure of the protein. Then it's going to take us through uh, information about the protein. So this includes the published paper about the protein. So it was published in 2006, and by this group of authors. And if you want to read about how it was characterized, then you can just click on this paper. But for today, we are just interested in the molecule. What's molecule? So the molecule is insulin like growth factor binding protein. So I'm just using it as an example. Then Homo sapiens, the sequence length 92. Then mutation is also very important to check the mutation. If we have zero mutation, then the gene name, if you want to look at the gene name, you can just click on this and it will take you to this. Then so uh so if you scroll down, you'll see a couple of things about the protein as well. Uh, so here it is the insulin. This is the IGF protein, so which is in complex with the insulin, right? The insulin receptor. So insulin like growth factors. So the first chain is the uh, insulin like the IGF uh, factor binding protein. Then the second chain. So we have two chain because they're in complex with each other. So both are proteins. So you can find information about the complex here as well. So before I move on to other database, let me just see if I can also show you another one. I think if so I have one protein in mind, which is uh, glycogen synthase, GSK, GSK. Okay. So which is GSK3 beta. So GSK3 beta has also been shown like dysregulation of uh like GS dysregulation of GSK3 beta has been shown to be involved in the increase in the like in the progression of diabetes. So for example, if I want to work on GSK3 beta, so I can just click on no, type it here, or we can go to Google search box and type in silico uh, JSK3 beta. So we just want to look at couple of papers that have worked on GSK3 beta using GSK3 beta using in silico studies. So like this molecular. So in this case, you just want to like find out the type of protein they use, like the name of the molecule they use for the GSK3 beta. So another way to go about it is you can just go to the materials and method, look at the crystal structure of the PDP ID of the GSK3 beta that were used. So in this case, they use uh, they use okay, they use two GSK3 beta crystal structure. So one is in complex with imidazole like an inhibitor. So we can copy this and we can just try to find out what type of molecule is it. So we can just paste the ID, okay, 6 5. So we can place the copy and paste the ID here, the PDP ID, and it's going to take us to the information about the protein. So so for the so this is particularly important when you don't really know, like you don't have enough information about the protein. So it's kind of important if you are just working, you don't really have enough experience with on how to utilize molecular docking. So it is kind of important for you to look at literatures that have worked on the same proteins and try to identify the proteins that they've used. So in this for example, this protein, this PDPID, you can see the name, the crystal structure of GSK3 beta in complex with a molecule. So this molecule defines the active site of the protein. So whenever you want to download, you make sure that the protein you are downloading has a small molecule that was complex with it during um, crystallization. So this is one. So you can just so let's look at the basic information about this protein. So it contains the two chains, so chain A and chain B. We have 3650 sequence length, then mutation 0. Then also, okay, so it contains two ligands. So the first one is okay, the first one is this, which is the inhibitor. Like it is evidence based on the structure, right? So the imidazole compound, imidazole compound, so derivative. So then the second one is it can be a cofactor. So you can see it is an ion, acetate ion. So it might be so important for the function of the protein. So so in this case, we already identified that, found out that this is the structure of the protein. So when you want to start your molecular docking, you must first of all identify where the binding pocket of the protein is by locating these compounds. So then after that, after you've peruse whatever is here, so the next thing is you have to download the molecule. So you scroll up, then you click on download files. So we are interested in the PDP format, which is this. If you are using um, Linux, I think you are going to use this. Yeah, Linux, you are going to use it like Ubuntu and the like, you are going to use this. Then the faster sequence is just like the sequence of the protein represented by alphabet like letters. So like amino acid, the first 20 amino acid as alphabetic representation. So so in this case, we can just see, let me just show you what it looks like. Uh, what it looks like. Uh, uh, Okay, so something of this. So you can see that this is the pasta sequence, just the amino acid of the protein. So that's all for this section. So the next section, let me just quickly go to the slide again. So we are moving next is we are moving to the compound database. So compound database, so we have the PubChem, right? So the PubChem. So to access PubChem, just type PubChem and click enter. So the first entry is the uh, homepage to PubChem. Then okay, so we've gotten to PubChem. So the next thing is, I said we are using, I assume that I'm using insulin receptor. So I need a standard. So, for every experiment, you need a standard, positive control, negative control. So for the standard, so I'm going to use metformin. So here you can search for any compound you like you want to use, any compound, uh, just type it, it's going to give you series of compounds, similar compounds related to what you type. So I will start for me. So because it's the first line of drug against diabetes. So metformin. So I type metformin and hit enter, right? Metformin. So it's going to take us to another page where we can, uh, where it's going to show us the series of compounds that are similar to what we type. So the best match. So it's always advisable to just go with the best match. So the best match is the best is the is the compound that match the name of what you type. So in this case, metformin. So metformin, you can see out of gives out about 60 different results. This one is melamine, melamine. So just go with the best compound, right? The best match, which is metformin. So metformin, uh, uh, just we click on metformin, then it's going to take us to another page. So here we can see a lot of information about metformin, the structure, the IUPAC name, everything. But for this particular section, we are not interested in that. We are just interested in the structure and how to download. So the next thing is we have to download. So click on download. So it's going to display a lot of things, that a lot of formats. So what format are we interested in? So it depends on what you want, but for the sake of molecular docking, just go with the STF. So you click on, we need the 2D structure because we have to prepare. So if you don't have a computational tool to explore, like to uh to prepare the compound, and you can just download the 3D structure directly. So but I advise you use the 2D structure, then you click on save, then you download. So this is how you are going to do for every other compound you are going to use. So for uh for another one, which is routine. So routine. So, so so let's just click enter. So routine is another bioactive compounds with huge molecular weight that has been found in numerous African medicinal plants. So routine. So we always go with the best match. So which is this routine. Then just click on the structure again. Then so this how you're going to do for every compound that you want to retrieve from this repository. So in case now there are cases where you identify your compound, like you characterize your compound using GCMS or SCMS. So and uh, for example, there are cases where you characterize. So for example, uh, let me just quickly show you something. I want to just show you like another way that so like there are cases where we identify your compounds using like SCMS, and you may not be able to, okay, like okay, I want to, like, you identify your compound using GCMS, and you want to like download the 3D structure of the compound, but unfortunately you didn't find the compound on the PubChem also. So for example, I just want to show you a paper. Okay, like in this case, you can see that in this paper, the authors use GCMS and SMS to uh, uh, sorry, okay, GCMS and HPLC to uh, characterize compounds, like identify compounds from a phytochemicals. So so in this case, let's pick one compound. So these are the list of compounds that were identified from the plants. So let me just find a compound that I think is not going to be on. Okay, let me pick this. Yeah, so let me pick this. So uh let's search for this compound on PubChem. So compound. Oh, fortunately we could identify it. So anyways, in some cases you won't be able to find out the compounds in PubChem. So in that scenario, you can use Chem Spider. So Chem Spider, so which is another compound database. So you use Chem Spider and you just the same in a similar interface. So you the same just paste the same name of compound. If you can't find it on PubChem and type and hit enter. So yeah, it's able. So in some cases, you will not be able to find those compounds in both database. So in that case, what are we going to do? We are just going to draw the structure using like Marvin sketch or Chem sketch. So I believe maybe this section might be taken by Mr. Abayomi. So that's all for the compound database. So next, let's move to, so next,
Opeyemi, please try rounding up soon.
okay, okay, thank you. So next, let's move to other database uh, like NPass. Then let me just take you through the ADMESA, which is going to be the last database that I'm going to introduce to you today. So ADMESA. So just type ADMESA on Google search box and hit enter. So I think I prefer the ADMESA 2. So just let's click ADMESA 2, which is the second version of ADMESA. So in this case, what we need to do is if we want to predict the ADMESA, which includes the ADMET and the toxicity toxic profile of the compound. So what you need to do is just copy and paste the smiles of the compound. So how do you get the smile? You can get the smiles of the compound from PubChem. So or yeah, from PubChem. So for example, so let me just go back to PubChem. Uh, okay, so you can see once you enter the name of the compound, you are going to see uh, it's going to show you information about the compound. Then you can scroll down and find the uh, canonical smile, which is this, which canonical smile is just the linear representation of the compound. So just copy this and you go to add method, then you just paste it, then you say it's to predict. So we are predicting using the not the old version but the new version. So predict. So or we can also draw the structure, but it's not advisable to draw the structure because sometimes we might mistakenly misdraw the structure. So yeah, so after predicting it and we click enter, it's going to show us the predict the admit property of the compound, so which includes this as well. So a lot of properties pertaining to the compound. So you can use this and put it in your manuscript for publication. So we also have Swiss ADME as well. So it's also serves the same function, except for the fact that in Swiss ADME you cannot predict uh, the toxicological property of the compound. So here what you need is just you can draw, but advise to always copy and just go and look for the smile from the PubChem and and paste and run. So for example, let's fill with example. So let's use one and this is ibuprofen. Then let's run. So yeah, so it's going to show us different properties about the compounds and whatever we need, either the physical chemical property, lipophilicity, water solubility, pharmacokinetics, and drug likeness. Either one, just depending on what you are interested in. So at this note, I'll be coming to the end of the practical section. So I appreciate everyone for taking time to, listen to this practical section. So, and if you have any question, you can ask. And also, I also apologize that I didn't prepare. I didn't make a very, uh, a very concise, a very concise PowerPoint for this section. I uh, forgot that today is my section, so I thought it was on Monday. Uh, so on this note, if you have any question, just you can ask me. So thank you.
Thank you so much, Mr. Opeyemi, for that presentation. Thank you for taking us through the practical aspect of the different data, protein databases. And yeah, please, if you have questions, either you type it in chat box or you unmute yourself. We have limited time, so we can take 1 or 2 questions, or we have comments. We have a comment on the chat. Hi, Dr. Florence, well done. Thank you for your presentation. Please, any questions? Okay, we have someone raising his hand. Mr. Ibrahim, please, you can go on.
Good evening. Thank you for the presentation. I really enjoyed the presentation. My question goes to the part when we are doing the admin, the properties, whatever. So what I want to ask in quotes is that what if we have more than one compound? Because I can remember sometimes if you have to dock like 200 compounds and you have selected like 30 or 40 compounds, you want to use your their property to streamline them before you go to another stage of analysis. So if you have like 30 compounds, are we going to be doing it one by one to screen their property, or is it possible for us to put them in batch in?
Oh, okay, thank you for your question. So it depends on the database. For example, the Swiss ADME can compute as much as 10 compounds at once. But the one of the limitation of Swiss ADME is you can't calculate the toxicity. So what I used to do is I use Swiss ADME for the normal ADME properties and ADMESA for the toxicity study. So for the ADMESA, you have to input the canoic casmide one after the other. So before, so you have to get the results one after the other. But for other database like Swiss ADME, you can just get as much as you can get as much as 10 information, like 10, you can input as much as 10 compounds at a go. So it depends on the database. I believe I have answered your question.
Thank you. We have someone else who wants to ask a question. Please go ahead, Mr. Muyiwa.
Hello, please, can you hear me?
Yeah, sure, I can.
Yeah.
Yeah, please. I just want to ask if we can have access to the recorded materials or the slides, because some of us came in late and we've actually missed a whole lot of the training, sir.
Yeah, you'll be able to get them.
All right.
Okay. All right, I guess that will be all for this part of the session. Thank you so much, Mr. Opeyemi Iwaloye, for your presentation. I'll be handing over to the next anchor for the next part of the session, Emmanuel Oluwarotimi. Please take over.